All right, hey guys, Mr. Brotherton here. So we are going to be talking about the structure of DNA today. So yesterday we talked a little bit about bioethics and some things that have come up in our society because of DNA. So let's talk about how this one little molecule can be so powerful. And it really goes to one of the biggest things in all of biology, which is the idea that the way something is shaped determines how it acts. So we call this structure dictates function. So what is it about the structure of DNA that allows it to make proteins, or in the case of a virus, allow a virus to hijack your machinery to make proteins, right? So the first thing, DNA is a nucleic acid. All right, this is a macromolecule. This is one of our four macromolecules we talked about early in the year. Okay, you guys uh, didn't know that they were called this, but you already knew what they were. Because if you look at the back of a label for food, that's going to tell you the amount of macromolecules that are in it. So if you remember, those are protein, they're fat, they are carbohydrates, and then the last one that they never list is nucleic acids. So it is a nucleic acid, which is a type of macromolecule that contains our genetic material. All right. Embedded in that molecule are really important sections that we call genes. These are the parts that have the instructions for making one single protein. All a gene is a piece of DNA that says, hey, this is how you make a certain protein. So proteins, extremely, extremely important. These are the little worker bees of your cells. They basically carry out all of your cellular activity. They carry out all of your cellular functions. They're really what gets stuff done for your body. Now, there's two different types of nucleic acids. There's the one we're really familiar with, that would be DNA, and then there's the one we're probably a little less familiar with, and that would be RNA. All right, now RNA, very, very similar to DNA. It just has a slightly different sugar, and the reason RNA exists and DNA exists is because RNA is a little bit older. We think that RNA was probably one of the first biological molecules on planet Earth, um, and so it's still kicking around. DNA is kind of like the advanced version of RNA. That's how I kind of think about it. I think about DNA as kind of, it could be Fruity Pebbles or something, and then RNA is kind of the off-brand version of that. All right, it's Tasty Rocks or something. That's how I think about it. So, uh, what is a molecule of DNA made of? Well, it's made of something known as a nucleotide. So a nucleic acid can be further broken up into these smaller little pieces called nucleotides. So a nucleotide, this is the monomer of a nucleic acid. That word seems scary, but it's not. All right, like a lot of words in biology, it tells you exactly what it means in its name. So mer refers to number, mono tells you how many numbers. So mer, number, mono means one. So it's basically one part of a nucleic acid. So your monomer is the smallest part of something, and then the larger thing, when we stick a whole bunch of these little monomers together, you end up with something known as a polymer, because poly means a lot. So just to summarize that really quick, a nucleic acid is a macromolecule, okay? It is a macromolecule made up of a bunch of little pieces known as nucleotides. That is a monomer. You stick enough monomers together, you end up with a polymer, right? You've probably heard those words before. So nucleotides, our monomer, our littlest piece of a nucleic acid, has three parts. It has a sugar, it has a phosphate group, and it has a nitrogenous base or a nitrogen base. So we have our sugar here, deoxyribose in the case of DNA, ribose in the case of RNA. We have a little phosphate group, and we have a nitrogen base here. So there's a couple different nitrogenous bases. One's called adenine, one's called guanine, one's called cytosine, one called thymine. That's only in DNA. And then in RNA, it has a different one called uracil. So in RNA, the thymine gets replaced with uracil. You guys have, oops, let me go back. You guys have probably seen the structure before. This should look very, very similar to our good old friend ATP. Okay, ATP looks pretty much exactly the same, except it has how many phosphate groups? It has 3 phosphate groups. It has one here like this, and then it have one here and one here. Remember, like I said, nucleotides, nucleic acids have been around for a very, very, very long time. Guess what? They can also be used for energy. ATP is actually just a nucleotide that's super specialized for energy. Other nucleotides like DNA and RNA are super specialized to make up our genetic material. Very, very cool molecule. One of the coolest parts of biology. All right, so let's get into the structure of DNA. So the first model of DNA was made by James Watson and Francis Crick. You've probably heard of those guys, but really it was based on images taken by this woman, Rosalind Franklin. She made these things called X-ray crystallographs. I'll show you a picture of them here in a second. And we'll kind of get into their story really quick. So you've probably heard this before. DNA is shaped like a double helix, which is basically just a twisty ladder. So if you look here, so here you have your twisted up DNA, and so it's all twisted like this, and then over there, right over here, you have it untwisted, so it's nice and flattened. Okay, so one, it's all twisted up, the other one, it's nice and flat. So you can see here is DNA has the sugar phosphate backbone. So phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then you can see that our nitrogenous bases are here in the middle. All right. So what's really, really awesome about, oops, let's go back. What's so awesome about this molecule is because of its shape, it is so good at its job, and it comes back to really basic chemistry. All right, so these bonds here in our backbone, pretty strong. It supports everything, so you really need a strong bond there. All right, so these bonds right here, this bond right here, this bond right here, this bond right here, those are all covalent bonds. So that's a bond where atoms actually overlap each other and they kind of share their electrons. So it's like that, nice and strong and overlapping each other. And since the way DNA works is something needs to come in here and actually read these little bases. Remember yesterday I said these are kind of just like a sentence, okay? Because something actually needs to get in here and read this and kind of unzip this. These bonds in the middle are actually really weak hydrogen bonds. So they're very easily broken. It's very easy for something to come in here and start reading these nitrogenous bases. It's one of the coolest things ever. In class, we usually model it with our bodies, where we kind of will lock arms, and then that'll be our covalent bond, and then we'll stick our arms out in front of us like that, and that will be kind of representing the hydrogen bonds, but obviously we're all sitting alone at our houses. So really quickly, I just wanted to show you Rosalind Franklin and the original model that Watson and Crick came up with. So it's kind of amazing because she was just taking these images where she would shoot an X-ray through DNA, and what would come out would be this kind of refracted or diffracted image. The way when you look at something through water, you know how it's kind of all weird looking and not perfect. Well, that's the same thing that's happening here. She would basically shoot light through these, and then it would kind of scatter, And what she was able to do through a whole lot of math, a crazy amount of math, was start to come up with an idea of what this thing looked like. Uh, she was a very serious bench scientist, which means she didn't really want to jump to any conclusions. She was working very slowly and wanted to kind of be super meticulous and make sure everything was 100% correct. She was working with this other guy, Watkins. They didn't get along at all. Rosalind Franklin kind of had a reputation for not being the easiest person to work with because she was so intense about what she was doing. And Watkins actually kind of smuggled her images out and gave it to these two guys. And they jumped to some conclusions. They happened to be right because they were pretty good scientists about what DNA looked like. So they didn't discover DNA, they just made the first model that accurately showed what it looked like. There were people, Avery, Chase, Griffith, all these other people who already knew what was happening a little bit with DNA, but these two guys were the first ones to really um, make a model of what it looked like, and it was pretty accurate. These two guys and Watkins ended up winning a Nobel Prize for that. Unfortunately, Rosalind Franklin had actually already died by the time they were awarded the Nobel Prize. Probably she got cancer, probably from spending years unprotected from X-rays. It's kind of sad. So a lot of people think it's a little bit of a sexist story because she really knew what was going on and never got the Nobel Prize. The issue is the Nobel Committee does not award prizes to people that are already dead. She probably would have actually got the prize along with the other three guys. Okay, so back to the structure of DNA. So these nitrogen bases, they can only bond with their complement. Their complement. So complement with an I. If I say, hey, that's a nice new haircut you got there, that's compliment with an I. Okay, that means to say something nice about someone. Compliment here with an E basically means the opposite of something, or kind of the match that goes with something. So like your girlfriend or boyfriend would be your complement. Okay, you guys kind of match up. So uh, the A's always bond with the T's, okay? The C's here always bond with the G's. And we call these the complementary base pairing rules. That's the way it's always going to be. They always line up that way. And if they don't, it can cause serious, serious problems. So let's go through and try and label our DNA here quickly. All right, so what is this thing? You've seen it before. They're always represented as these little circles. You saw them in ATP. They were the P part of ATP, and that would be your phosphate. So what's this thing? That would be your deoxyribose. That is your sugar. So these together make up our sugar phosphate backbone. What is this thing? What is this bond representing? Remember, this is a strong bond. You want your backbone to be strong. That is a covalent bond. All right, so what's happening in here? This thing gets unzipped, all right? So does that bond need to be strong or weak? It needs to be weak. That is our weak hydrogen bond. And these three things together, our phosphate, our sugar, and our nitrogenous base, this whole thing together, what is that called? That is called a nucleotide. All right. So let's talk about RNA really quick. RNA, like I said, this thing is super, super old. It's been around forever. It has some differences to DNA. So biggest thing, it's a single strand instead of a double strand. If you look here, it doesn't have a complementary base that kind of lines up with it. It's just hanging out all by itself. So it's a single strand. Your RNA bases do bind with DNA bases. They're just a little bit different. Okay, so DNA makes RNA. Eventually, RNA goes out of a nucleus and is used to make proteins. So your A's, they bind with your U's. Your C's, they bind with your G's. So it's a little bit different. These guys have uracil instead of thymine. So real quick, our bases in DNA are A, T, C, and G. In RNA, they are A, U, C, and G. Deoxyribose versus ribose. Our shape here is a double helix versus a single strand.